Okay, so this is probably within the first 90 seconds or so. But after those 90 seconds, cellular respiration is the only way that we can continue generating enough ATP needed for you to keep exercising, for you to keep running. So immediately we are having lactic acid fermentation build up inside of us. But after that point, about 90 seconds or so after that point, cell respiration kicks in. We enter into the Krebs cycle, we enter into the electron transport chain. And this makes sense. Why should our bodies spend all this time trying to use up oxygen if you're just going to run, a 200 meter, if you're just going to run a little bit, or if you're just going to have fight or flight. It's not going to waste its time trying to go into this real detailed process of getting ATP. But after 90 seconds or so, it will spend the time because the body is going to realize, your cells are going to realize, okay, we're doing some hard work. We need some energy. Stat. Okay, so let's talk about the Krebs cycle. The Krebs cycle, we've gotten pyruvic acid that came in from glycolysis, and it's going to be broken down into carbon dioxide in a series of energy extracting reactions. You can think of cell respiration as a way to extract energy that's stored within a glucose molecule. All right, so this is also called the citric acid cycle, and it's because the first compound is citric acid that is formed within the series of reactions. Okay, so pyruvic acid is from the glycolysis, and it's going to enter into the matrix. The matrix is the innermost part of the mitochondria. So remember back to your organelle stuff, the matrix is this inner portion right here, this darker portion all along this area. So pyruvic acid enters in, and one of those pyruvic acids is in the mitochondrial matrix, and an NAD plus will accept two high energy electrons to form NADH. So that's happening right over here, and that's happening when carbon dioxide is released. Okay, so right there. I could have just left it. Oh well. Okay, so the remaining two carbon atoms that are left from pyruvic acid. So notice, let's go back to this right here. We had 3 carbon pyruvic acid, 1 carbon left so we could form CO2. Now we're left with 2 carbons. Okay? Those are going to react and they form what is called acetyl-CoA. Acetyl-CoA, don't worry about the real long name, acetyl-CoA is the 2 carbon atoms that are left before it really fully enters into that Krebs cycle. Okay, so energy extraction, let's get some energy. We have citric acid that's going to be formed. So after, what's really happening is we have a 4 carbon compound over here forming with acetyl-CoA, and we get citric acid. Okay, that 6 carbon citric acid is going to break down into a 5 carbon and then a 4 carbon compound. Okay? And with that, 2 carbon dioxides are released. 5 carbon into a 4 carbon. Notice, here's the 4 carbon. Okay? And then at that point, we're done with this molecule. It can cycle back and get put together with another acetyl-CoA. So this cycles back all the way here so that it can be used again. Okay? So energy will be released when we are breaking and rearranging all those bonds to get that 4 carbon molecule back. So as we are going through this entire cycle, we are getting ATP, FAD turned into FADH2. And FADH2, don't freak out, don't worry about it. It's just another type of electron carrier. It's another brand of electron carriers, okay? So now we've got ATP that's carrying some energy, and then NADH and FADH2, those are electron carriers. So they're actually going to get shuttled over to the electron transport chain. So there's our NADH, those are our NAD pluses, and then FAD are the electron carriers, and NADH and FADH2 will be used in the electron transport chain to generate ATP. So all of those molecules are going to get shuttled down to the electron transport chain. So you can think of the Krebs cycle as a generator of these carriers. Okay, we got some carbon dioxide, but that's a waste. That is needed to break down these 5 and 4 carbon molecules. We need those things to occur. But really what's happening is we want this, right? We need this because in the Krebs cycle we're only getting 2 ATP. But it's the electron transport chain where we're actually going to get lots and lots of energy, so we want to have electron carriers so that we can do that. Okay, so let's look at the final count at this point. We've already put in 1 glucose and we get 2 pyruvic acid from that. This goes into the Krebs cycle, and this will actually turn twice. We have 2 pyruvic acids, so we get 2 turns of the Krebs cycle, and that's how we will get our 2 ATP. So at the end of the Krebs cycle, after we've made 2 complete turns, we will get 6 carbon dioxide molecules, which matches up with our cell respiration equation. 2 ATP, still not enough to do, to blink your eyes a couple times. We'll get 8 NADHs, that's a lot of electron carriers, and 2 FADH2 molecules that will get sent to the electron transport chain. Now, NADH and FADH2 will pass their high energy electrons to electron carrier proteins in the electron transport chain. So now we've moved into the electron transport chain. So we are still in the matrix, but look, now we are right along the inner mitochondrial membrane. Okay, and the matrix is here. And then we have the intermembrane space, So this is like the stroma from photosynthesis. Okay, the matrix is like the thylakoid's space from the thylakoid. Okay, so at this point, those electron carriers, NADH and FADH2, we break a bond. When we break a bond, we're going to break hydrogens off and get some electrons to pass through. Those electrons will go through this entire electron transport chain, and at the end they combine with hydrogen. Okay, so this yellow line right here, these are protein carriers. This big one right here is a protein carrier. This one over here is a protein carrier. Okay, now where do you think we're getting these hydrogen molecules? They are coming from the breaking of NADH and FADH2. So those NADH molecules will break off hydrogen. They'll stay within the matrix. Okay, and now at this point, these electrons will get together with the hydrogen and the oxygen so that we can produce water. Now, energy from the electron transport chain is used to move hydrogen ions against a concentration gradient, right? It's just like photosynthesis. When that electron passed that tunnel-looking protein carrier, hydrogens could get pumped through. Remember when we practiced outside and we had the tunnel, the 2 people made that tunnel and hydrogens walked through every time an electron passed through. The same exact thing. But we are going, instead of from the stroma to the thylakoid space, we are going from the inner membrane space into the matrix. Okay, now we will get I think I just totally told you backwards. I told you backwards. Hold on. Erase what I just said to you. Okay. They're pumping this way. Sorry. They're pumping from the matrix to the internal membrane space. I'm really sorry that I just told you wrong. I'm sorry. And I'm totally not starting this video over because we are well into it and I can't talk anymore. Okay? Okay. Now, if you notice, we have a high concentration of hydrogen ions in the internal membrane space. Tons of hydrogens, right? We still have Mr. ATP synthase over here. And what's going to happen? We have lots of hydrogen molecules, all these little things right here, right? They're going to pump through. It spins, same thing like photosynthesis, and we get a production of ATP. Just like photosynthesis. Okay? So here is a close-up picture. It's from an updated version of your textbook. It's a good-looking picture, but you can see the entire process. You've got your NADH, NADH and FADH2 that came from the Krebs cycle. Those molecules are going to get broken off. You get a hydrogen molecule that's going to hang out in the matrix. Okay? Now, as that's happening, you've got this yellow line that's representing electrons coming from the NADH and the FADH2. They're going to go through that electron transport chain, through those electron carriers, and go up into the matrix and meet with hydrogen molecules that are hanging around in the matrix, some oxygen, which is what we breathe in, and those electrons will meet and form water. In the meantime, we have hydrogens that are going to pump through the electron carriers every time an electron passes. And then look, we have tons of hydrogens all over the intermembrane space. So, high concentration, not comfortable. It's going to go through the ATP synthase. The ATP synthase moves, it spins, and we get a production of ATP. So, how much energy are we really talking about here? Okay, one glucose will break down and we get a total of 36 ATP. Okay, we get a total of 36. We have 2 from glycolysis, 2 from the Krebs cycle, and 32 from ATP. 32. So the total is 36. Now, this represents about 36% of the total energy that's in glucose. So the remaining energy is actually lost. So 64% of glucose is actually lost as heat. And that's just something that naturally happens. It's not as efficient, but when you compare it to photosynthesis, cell respiration with the use of oxygen is quite efficient. To get 36 ATP molecules total, that's a lot of energy. So that concludes Chapter 9. That concludes cell respiration. Please go back and watch this. Please draw pictures if you need to. Review the Krebs cycle. Just like the Calvin cycle, it's can be a little bit difficult. You've got to learn some names. But hopefully you guys will be able to apply what you know from photosynthesis carried over here. So if you have questions, email me, ask me in class, but I'm confident that you guys are going to understand this. Alright, I'll talk to you later.